I think so. Yeah. Is it? Did I not turn it on? Oh, it's good. Okay.
Okay, so now we're going to another track on fundamentals of bioinformatics. How is it like for a data scientist to work with biologists? And we've been able to do this work together with Michael from Inflammatix in the US and Dr. Helen Nigussie of Addis Ababa University, yeah, and myself. So I'll start with Michael taking you through the whole pipeline, the end-to-end thing of how it is in the using how it is like when using biological data. Okay, yeah, welcome, Michael.
Thanks, Dina. Let me see. There's full screen in here, right?
Just F5.
Oh yeah, that's right. Yeah. Sorry, I'm out of it right now. I think I did F5. Did I not do it? Ah, yeah. Here we go. Yeah, there we go. Sorry, I'm awake now. Right. So thank you very much for having me here. It's a great privilege, and I have to say, on a personal level, it's good to connect with colleagues in Africa. It doesn't happen that often, but I hope it happens a lot more in the future. Please forgive my voice. My 5-month-old son got sick, then my wife got sick, and now I got it. So I'll try to get through it, and I'll be brief too, so Dina and Helen can do their thing. So what we're going to discuss today is a particular technique for analyzing gene expression data called RNA-Seq, or which is short for RNA sequencing. So I'm just going to walk through a motivating example based on a freely available Jupyter notebook that Dina found that was looking at the Zika virus. And they did some RNA sequencing, and we'll kind of show end-to-end how this analysis goes and what it's good for. All right, so what is it? So I already said a bit of this. It's short for RNA sequencing, and it's a process of assessing or evaluating the expression of a gene across a genome by sequencing the RNA transcripts from a collection of cells. This is important to mention. What you're doing is you're taking some sample, you're digesting the cells, breaking them basically up so that the RNA is free floating, you're isolating it, stabilizing it, performing some chemistry on it, and then doing these assessments. So, you can do RNA-Seq then in any cell population, right? You could look at someone's liver, you could look at someone's blood. This is a very general technique, as long as you have a way of getting to those cells, digesting them, and doing this analysis. All right, so this is as general a schematic as I could find on how RNA-Seq works. And I am not a chemistry person, so I am not even going to go into a lot of how this works. I apologize if it's hard for people to read. I'll try to narrate it as I go. Never mind this in vivo, in vitro, in silico. It's kind of unnecessary. Really what matters is this flow. So you have a gene in the genome. You have a particular region, and perhaps you want to sequence across every gene in the entire genome. What you want to isolate are the mRNAs. But for sequencing, mRNA is highly volatile. It degrades rapidly, and so it's hard to work with. So generally, to get, say, once you've done this digestion of cells, to get those RNA sequences, what you do, well, first to make the sequencing faster is you break it up into fragments. This is inspired by, for anyone who knows how the whole DNA sequence was done, shotgun sequencing. So this is just where you blow apart the sequence. And then to make it stable, you do reverse transcription to get cDNA. That's this step here. So cDNA is, as its DNA, composed of the nucleotides of DNA, and so it's much more stable. You can leave it out on a counter for days, and it's not going to do anything. So it makes it much easier to work with. Now, this is the part where I'm going to get fuzzy because I don't know all the chemistry, and every company has their own way of doing it. But from these fragments, you then come up with a set of reads, sequencing reads. Those are these short bits here. So in that parlance, in RNA sequencing parlance, these reads are what you're going to use to digitize the expression of any gene. by aligning the sequences based on their composition to positions in the genome. So the idea is, you've isolated these sequences, they correspond to some region of the genome, you perform your sequencing, and now you align back to the genome to see, okay, well, here's where most of these reads are coming from. They're coming from gene X, Y, or Z. And this bit down at the bottom, I'll just say, a gene region doesn't always produce one form of a gene. So RNA sequencing is also useful if you're interested in the other forms. So these are called splice isoforms. So these are these variants here where in one gene, in this splice variant, you had 3 exons, so 3 protein coding bits. And in this one, you only had 2. So this is actually what makes the genome so interesting and effective, is that it's a kind of a fun aside that they can kind of treat their genome like a Swiss Army knife. They can modify their own genes, or at least have evolved to do this. So this is the basic flow. I'll break it down in another way, which is germane to what Dina is going to talk about in the tutorial. So this is really what the steps we're going to go through, and we'll kind of show some tools for how to do this. The first thing, as anyone who's worked with data would say, is you got to do quality control. So there's QC that happens at the top. We'll go through some tools for doing that. The reads mapping, that's the step I showed you on the last slide here, where you see these reads pile up, they're aligned to the genome, and you need this alignment. So you need a reference genome to do this alignment. That's sometimes tricky to get, especially if you're working with an organism, say, that isn't like us, that's highly sequenced. If you have like a brand new organism, reference genomes may be hard to come by. But let's assume you have it. The next step is just doing the quantification of expression. So it's not enough to just align the reads to the genome. You have to come up with some function that translates that kind of histogram over your gene into an actual value for how much that gene was expressed in your sample. And once you have that, you can start asking interesting questions. Let's say you're looking at a disease, and you want to say, well, is gene X more expressed in the disease versus normal, versus healthy? So you can use these genes then and their expression, which is what I do in my work, as, say, an indicator for a condition. What we actually look at is indications for infectious disease. And genes have been incredibly valuable and accurate in doing this. The last step is more maybe of an academic nature, but still interesting, and it's becoming more important now for pharmaceuticals. Say, looking at genes that are highly expressed or lowly expressed, and asking whether they're enriched for a given process, say cancer or cell division, or whether they're enriched for targets of drugs. So knowing, okay, this gene is highly expressed, but I have a drug for that, so I can knock that down to help maybe mitigate the condition I'm seeing. Any questions so far? Great. Please stop me if I get on a roll. So this is actually the exact, and this is the last schematic I'll show, I promise. This is the exact schematic for the notebook that Dina's worked through, and I've annotated it here with these different colored boxes to correspond to what you just saw on the last slide. So here, just for more information, are some of the tools you can use to do QC. So there's fast QC we'll look at for quality control. Read mapping can be done with the STAR tool, which is very common. Expression quantification and normalization, you can use these tools here, EdgeR and feature counts. The authors had kind of a pet tool that they liked for differential expression analysis called characteristic directions. We'll look at that. And then there are these libraries down here for functional enrichment. Yes, Ernest.
So the reads mapping, why is that important? Why can't you just take apply it directly to a model? Why do you need the reads mapping?
Yeah, so you need to have a rough idea of what genes the reads are actually aligning to, so that you know that the reads as measures of the transcripts are corresponding to that gene. So I guess a better way of saying it is that, let's say I sequenced 10 transcripts, and 5 of them align to this gene, 3 of them align to that one, 2 to this one. I could use that then as a proxy for saying that I have expression count 5 for gene X and expression count 3. But on their own, I wouldn't know what those reads corresponded to in the genome without having it. So I'd have to do that alignment first to know this gene has this many expression units.
And if you're doing a task like maybe prediction or classification or something like that, where you're just taking replicate and predict the value, for example, would you need to know the actual genes?
Yeah, because the genes themselves are actionable in a clinical sense, or they're useful for knowing what drug do I need to make, because drugs can be made to bind to particular proteins or targets, right? Or if a particular gene is up or down, that may have for example, in our work, that may have an impact on whether that person is infected or not. But not knowing whether it's a gene, just having kind of the reads without knowing where they go, it may not have enough information. That extra level of annotation gives us something more. It's a good point, though. All right. So the first stage here, we're looking at the quality control of the raw reads, and there's so many ways you can look at sequencing quality. I'm just not going to go through all of them. This is just a slice of it here. You can look at per base quality, per read quality, whether the nucleotide composition in the read looks right, things like that. And the FastQC, the tool we're going to look at, actually gives this to you, all of these measures. So you get a very good sense very quickly for whether your read data is useful. So here what you're looking at is the position in the read in terms of the base pair number. And then you're looking at the per base quality along the top. So this, you can clearly see this is great sequence in terms of per base quality. The thing to keep in mind with all these tools is they have little caveats. So FastQC was actually designed for DNA data. And so it kind of chokes. You see there's this failure measure here. It chokes on data that it wasn't expecting, but that doesn't mean it's bad. So RNA sequencing data is actually fine. It just doesn't like it, at least for that particular measure. So as I was saying before, and as Ernest pointed out, why do we need to do this alignment? It helps us to do the quantitation so that we can assign to any gene a digital count of expression. So it's really like building a histogram on the genome. And now you need to do some kind of smoothing or averaging, and then you have to assign a number to that gene. So these really are kind of discrete counts. There were this many sequencing reads that overlap this position. That is the next stage here, this mapping. So once you've done the mapping, you need excuse me, some measure of how much expression you got per gene. Well, that can depend on a couple of things, and so you have to normalize appropriately. You can't just take the reads as is and where they line up. You also have to consider, well, I might get more reads because my gene is longer, so there's more chances for me to hit it with reads. Or I might get more reads because this was a particularly rich collection of cells I got, and so there's just tons of reads everywhere. So I got lots of reads everywhere. So I need to be able to normalize now with respect to gene length, as well as the total amount of reads I got in my sequencing run. So that's what these two measures here I'm showing, CPM and RPKM, do. They just do the normalization in different orders. So CPM is just transcripts or counts per million, where you first normalize the read counts for any region by how long the gene is. Then you do a summation here of these RPK values, divide by 1,000,000, that's your per million scaling factor, and then you get your CPM by dividing each of these normalized values, these RPKs, by that overall scaling factor. Now RPKM flips the order a little bit, but it's just important to note here that you have to do this normalization. Just the raw sequencing read counts don't help. Any questions so far on this? Sweet. Okay. So now we've got our expression data, and we have to ask the really big questions, right? Because now the data science begins. We've done all the quality control. We slogged through all the pre-analysis. Now we need to know what patterns are in our data. What's the structure? A common question when you're doing gene expression analysis is, should I consider every gene? Because for one reason or another, either because in your condition that gene's not expressed, or because the run just didn't pick up any expression, there's going to be genes with little or no expression. So you don't want to really pass these through a forward analysis. So there's going to be some filtering, some pruning. The second, and I would argue more important question is, say if you're looking at multiple conditions, like I often do, are expression differences you see, so differences in expression from, say, disease to healthy, down just to the condition I'm looking at, Or is there some other kind of skeleton in the closet, something I need to consider that's making me think that expression is different when it's really not? And that's what's getting shown here in this PCA plot. So here are the first two dimensions. And what they're colored by, so you have these orange dots down here. This is a particular sequencing platform, MySeq. And then in blue on the top is a different one, so completely different technical platform. But they should be generating the same results, right? And they're not, right? There's clearly structure. There's clearly clustering by platform. So that means that there is differences in gene expression that have nothing to do with the conditions I'm interested in. And this is called batch effects, more generally. These are just a major bugaboo in the gene expression analysis community. They can make it very hard to pool data from different platforms and different settings, which then keeps our sample sizes low, which makes data science in the medical field particularly very hard, as some people here may attest to.
So you did a PCA on
Yep.
So what is the correlation between those genes and not linear, for example?
Oh,
you did a PCA, so how would you?
Yeah, that's a fair point. Yeah, you could use other embedding procedures. There are plenty out there now. In fact, there's kind of a small holy word developing on who should use what. There's T-stochastic neighbor embedding, UMAP.
So the reason I ask is, I don't know, but these may influence the trust model.
Oh, yeah.
Yeah, so I don't know if it's better to just assume that there are nonlinear correlation between these points and then get a picture.
It's a fair point. I feel like the PCA is easy. And so if you can see the structure right away, it's cheap, it's implemented in a lot of different languages, then you know. But I agree, if you don't know, that doesn't mean it's not there. So you might want to try another embedding. But then if you don't see it with a nonlinear embedding, it's probably not there. Yeah, that's a good point. Any other questions or comments? This is just another view of the same issue. So here you've lined up the 8 samples, Zika infected, and then mock. Really, this is just non-infected. And you can clearly see, this is clustering by gene. You can clearly see structure. So differences here between Zika and mock, differences here between Zika and mock. Good, right? So for these genes, there's clearly differences by condition. But for these genes, they seem to be different with respect to the platform that was used. So just using those genes to try to do an analysis to say, predict Zika might be more difficult. You'd have to account for the fact that, what you did to get that expression matters.
So is that different because of some chemistry thing going on in the way they do the actual work?
It's possible, yeah. I with batch effects, at least I've always looked at it like, well, I guess we're stuck with it. And then you try to adjust afterwards. But yeah, it could be chemistry, could be, there was a batch effect I heard one day that blew my mind. A guy was doing protein isolations in a lab down the hall from me at McGill. And he kept getting this weird issue where his values were always lower on a Friday. And it was because there were fewer people in the lab. And so the air conditioning was shut off. And so the humidity was different. And because once they turned the air conditioning back on, the values were stable. So just all kinds of issues like this need to be borne in mind. And so data is not always pure data, right? Right, so we've got our expression. A next question we might want to ask, a very key one, is are there genes that are differentially expressed across conditions? As I said, these can be useful as markers for the disease or for the condition you're interested in. So here's just a smattering. This is a listing of genes that came out of the analysis from the different platforms. You could see they're less expressed. So these might be useful, say, in separating. Once we have these genes with high or low expression, you can take those lists and basically use tools to do enrichment analysis. So there are libraries like KEGG. There's this MGI mammalian phenotype that can help you get some sense for what those genes are doing in your condition. So what's interesting here is that genes with high expression in Zika infected samples are enriched for this prenatal lethality phenotype in mice, which as some of you may know, in Zika is a common phenotype. You get that microencephalopathy. All right, so I'm not going to say much more, just a completely unsolicited advertisement. I don't even work for these guys, but I figure this is the right audience for it. So the African Society of Computational Biology and Bioinformatics has their annual conference in Kumasi this year in Ghana. Submissions are still open for oral and posters, so if anyone is interested in bioinformatics work of any nature and has something they want to show, this is a great forum for it. There's a few resources here. The tutorial, Dina will, I'm sure, let all of you know where to get the slides, but these are some of the resources I use. Great tutorials and video explanations here. And yeah, thanks for your attention again. I appreciate it.
Thanks a lot, Michael. I'm just going to take you through the pipeline that Michael has explained.